corner of the steel. This is a big look at the cortex. It's purple because the cells are staining because they're containing starch, and then here's the dermal tissue right in here. Pictures of the steel coming up. But again, here's the protoderm will eventually give rise to the epidermis, which will essentially elongate and then specialize into, here's root cells. And why is that? It greatly increases surface area and it absorbs minerals, especially water. Here's a really nice shot of the cortex right here in purple, which is ground tissue, and then the stele, which is the vascular bundle inside the root. The tissue that you see here in red, it sort of looks like a Y shape, is the xylem. This greenish tissue over here is stained, which is the phloem. And then this outer ring is called the endoderm, as opposed to epiderm. Epi is on top over on the outside. Endoderm makes up the outer portion of the stele. There's even a cell layer right below the endoderm called the pericycle. I'll come back to that a little bit later. And so this procambion, which is primary meristem in the apical meristem of the root, will eventually give rise to the stele, which is composed of vascular tissue, xylem, and phloem. Here's a cross of the xylem. Look how thick those cells are of the vessel elements, and those are because they have a secondary cell wall, sort of forms like cells radiating like this, and then the phloem cells are right over there. Notice here, do you see here in the endoderm? Do you notice how these cells are a little red right in here? They're red because they're producing this wax that surrounds them, called the Casparian strip. I'll come back to this later. It's really cool. It prevents fluid from passing into the stele that is not wanted. In other words, there could be some toxics in that fluid that's moving in between plant cells, and you don't want that to get into the stele, because this is sort of the elevator of the plant, if you think about it. And so I like to think of it as like you're entering a building. First thing is it's hard to get into the building because of security. For example, number one is the epidermis, and so it's not going to let anything in. And then if you do get past that, you can move through number two, which is the cortex, or ground tissue. You can sort of go in between cells. If you travel through a cell, you'll have to go through the membrane, and of course the cell membrane is selective, so if you're unwanted, you won't be able to get in. But you can pass on the outside of cells. But you're not going to pass through the number three, which is the stele. That inner layer right in here called the endodermis has that Casparian strip, which prevents that. And so this cross is the xylem, and then over here is the phloem. So vascular tissue, ground tissue, dermal tissue. This is the root, the dicot root. And the dicot root is known for this sort of cross shape. Here's the endodermis, the pericycle, which is this one cell layer underneath. Here's the xylem, here's the phloem. Here's the cortex, so it looks like it has the measles, which are little leukoplasts of starch. Found this picture. This one's really cool too. You can see here, this is a nice shot of the stele. Notice here you have your xylem here, and then just on the outside of that is this pericycle, and then just on the outside of that is the endoderm in purple, endodermis, right there. That's kind of neat. Here's the root cap, apical meristem. Real close up of this. Here's the endoderm. Let me highlight it. Here's the endoderm cells right here that go around. One could say that the endoderm is considered technically the innermost layer of the cortex. And then you have this pericycle. And it's like, Anne mentioned that before with the story. It's this cell layer just on the inside of the endoderm. And why it's significant, I'll just mention it now, is that these cells are capable of division. And if they're capable of division, what can happen is they can bust through the endoderm, and it can actually form a lateral root. When I say lateral root is when you have like branches coming like this off of roots. So how do those branches form? Well, they form when the pericycle becomes meristematic, and it starts to penetrate through the endoderm and through the cortex. And so there's endoderm, pericycle, phloem, and xylem. Notice how thick the xylem is because of the secondary cell wall. By the way, what cell type is this? It's sclerenchyma, secondary cell wall. So this is I like this picture a lot. This is a sort of comparing and contrasting what a dicot root looks like. A dicot root is what we've been looking at primarily. We've been looking at the xylem, which is across here. Here's the phloem, which is in between. Here's that pericycle I was just talking about it, and then the endoderm, and then the cortex, and then the epidermis. And you're like, yes. But then in the monocot, like for example in grass, fibrous roots of grass, what you're going to see is it's not going to be a cross in the center of the stele like that, but instead the xylem are going to be in these little bundles right in here, and the phloem is in between the bundles. And then you have this little region in the center called pith, which gives support. It's a type of ground tissue, parenchyma cells. Pith, which provides some support for the root. And then here it has cortex and epidermis. So take a look at the real monocot root. So here's the real monocot root. Dermal tissue, ground tissue, vascular tissue. And then notice how the big circles right here are the xylem. All these little cells in between are the phloem, and it has this pith area. Here's a picture of a branch emerging. Again, monocot roots. The xylem cells are a little thicker. You can see here, phloem is all in between. Right in here is the cortex towards the outside. The pith would be on the inside. Here's a real close-up of the endoderm right in here. Here's the pericycle of the cortex over on the side. If I were to ask you, like, what is that? You'd say, oh, clearly that is a cross-section of a root. It's clearly a dicot root. This is a close-up of the stele. You can notice here the endoderm is highlighted in red, and you have the pericycle, which is this one cell. Here's the phloem and the xylem, and it has the cross. Now, this is a great picture showing side-by-side dicot and monocot. So again, here's the cortex, here's the dermal tissue, here's the stele, xylem, phloem, etc. Then over here, dermal tissue, cortex, and then you can see the stele located here. You can see the endoderm is highlighted in red because it has that Casparian strip that stains a little darker. These bigger circles are the xylem. You see that? Right there, the bigger circles, and then the pit is on the inside, right there, and then here's the phloem. So I was alluding to this before, like if you're going to produce a lateral root, in other words, a root that's coming off on the side, you got to bust, it's like sort of breaking through, you got to bust through the epidermis, you got to emerge through the cortex. And so where does a lateral root originate? Where does it sort of spring from? It springs from this pericycle layer. Those cells become meristematic and they start dividing. Not only do they become meristematic, it's a primary meristem, so it's going to become protoderm, procambian, and ground meristem. So this root is going to have its own ground tissue, its own epidermis, and its own vascular stele right there. And so let's draw our attention now away from the root and look at the shoot. And so if we're talking about primary growth in the shoot or the stem, where's it going to be? It's going to be at the tippity top. So this is I don't know what this looks like to you, like some house right here with the door, but basically this is the top, the apical terminal bud of a shoot. So in other words, these are the cells of the apical meristem that are dividing, mitosis, mitosis, mitosis. And what do they make the three primary meristems same as in the root, thank goodness. So in other words, the protoderm will eventually become the epidermis of the shoot. The ground meristem will eventually become the ground tissue, and the procambian will eventually become the vascular tissue. And so if you take a look at this meristematic region, this apical meristem, you don't need a root cap, but this apical meristem up here on the very top of the plant are the cells that are dividing. And so, as I mentioned before, let me just say it again if you don't mind, just for clarity, the primary division of a plant for growth, primary growth in an upward direction, the reason it's going upward is because the cells are pushing down, so it's going up right here. What you got is protoderm, which is eventually going to become the dermis, procambian, which is eventually going to become the vascular tissue, and ground meristem, which is going to be the ground tissue of the root. So these are three primary meristems, and they're at the tippity top of the shoot. And so this is a stem. So if you take a plant, an herb, I know it's an herb, it's not a woody because it's not very thick, but if you cut a stem in half, this would be a typical monocot over here, and this would be a typical dicot over here. There's some differences. One of the things that you're looking at is this vascular tissue, and these vascular bundles. You notice here, these vascular bundles in a dicot are around the perimeter of the stem. This is, again, pithy area in the center, so they're around the perimeter like that, whereas look here in the monocot, the vascular bundles are sort of scattered throughout the stem. You see that? Now, you wish you had a higher magnification, and so yes, I agree. So here's the dicot, here's the pit in the center. What's the pit doing? It's providing some strength and some stability along with the cortex. The cortex is not so much a function in the root as storage, but it provides strength. And so the cortex, if you remember this, is made up of chlorenchyma cells, which have a little bit of a thicker primary cell wall. If you look real close at one vascular bundle, you'll notice how the xylem is more towards the center, and the phloem is a little bit more towards the outside or cortex. So you got dermal, ground, vascular, ground, right in here. And so this is a dicot. Again, the dicot, and here's the monocot. Notice the vascular bundles are sort of spread out in between. So dicot, monocot. Here's a close-up of the monocot stem. The monocot stem has these little vascular bundles, but what's interesting is botanists, I guess, trying to be funny, maybe, I call these vascular bundles little monkey faces because they look like they have eyes and a little bit of a nose, looking like a monkey. I don't know, maybe. Here's the eyes right here, the monocot stem. These are obviously the very large xylem vessels. Notice how thick they are. They carry water. Here's the phloem. I like this picture a lot. Look at the cyst tube members, which are also hollow, but then they have these little companion cells, which are really helping out right there. You know, this doesn't have a large amount of cytoplasm right there. So this is a close-up of a vascular bundle of the stem of the monocot. And then getting to the leaf, the leaf has this upper area right here, which is dermal, and then it has this ground tissue, which is called in the middle, or mesophyll. The vein is not shown in this picture. It has this waxy cuticle. And the epidermis, a single cell layer, sort of like a puzzle, is the first line of defense against pathogens, and also the cuticles to prevent water loss. And so what's fascinating, you might be familiar with the leaf from photosynthesis, but if you look on the underside of a leaf, you'll notice that there's these specialized cells that flank these pores called stoma. These specialized cells are called guard cells, and they're capable of photosynthesis and opening and closing. They swell open, which allows carbon dioxide to come in and oxygen to leave. And so here we have is the dermal tissue, and then the yellow is the ground tissue, and then we have more dermal tissue here. And where's the vascular tissue? The vascular tissue is right here. we call it a vein. And the vein is made up of surrounded by bundle sheath, if you remember that, the bundle sheath cells that surround the vein, and then inside we have the xylem, which conducts the water to the leaf, and the phloem, which conducts the sugar that's being produced in photosynthesis, dissolved in water, away from the leaf, down the petiole to the stem, and then ultimately the root, or it could be to a fruit. And so again, we'll talk later about how guard cells regulate transpirational losses, but they do. They close up at night and they open up in the morning generally. But this is where gas exchange takes place, like these little mouths right here. So gases go in and out, and also water goes in and out. Water leaving is a good thing, transpiration, because it pulls through capillary action, that's cohesion and adhesion, it pulls water up the plant, and that's how water is able to travel up xylem, is through transpiration. And so that's providing the pull, especially if it's windy, especially if it's sunny. You have a very low water potential in the air, so the water is able to leave. What's troublesome, though, is excessive transpirational losses can be lethal to the plant, and so that's a cause for concern. And so you can see here, dermal, there's ground, and vascular in the leaf. There's the stoma, the guard cells, which allow the gas exchange, oxygen out, CO2 in. And that's the mesocell, which is ground tissue. These are parenchyma cells, these are palisade, these are spongy in relationship to the way they're close to each other, like a palisade, like a fence, which captures a lot of sunlight. Spongy allows air circulation to occur in the leaf. So it's really a cool structure. This is one of my favorite organs of a plant, is the leaf. It's an incredible example of structure related to function. It's awesome. And then finally, the vascular tissue, I was mentioning this before, travels from the petiole into the leaf. Then it goes into, it brings water to the leaf for photosynthesis, because as you know, inside the plant cell, inside the palisade, the chloroplast needs water, especially in the thylakoid membrane, in the act of photosynthesis in order to take place. So there you go. Dermal, ground tissue, palisade, spongy, lower epidermis. Notice the lower epidermis has the guard cells. Notice these air spaces allow for circulation of gases. So I hope you enjoyed this discussion on primary growth in plants. Thanks for watching. Hi everyone, it's Mr. Cinti, and I'm happy today to be talking to you about plant hormones. That's right, plant hormones. We're not the only organisms that produce hormones, that is animals. Plants also make these hormones. They're chemicals that are capable of moving from cell to cell, and they communicate a message. So it's sort of like a chemical text message. And it's produced by one cell, and it's received by another. Usually a specific cell is destined to receive it, and then that cell does something. That cell either divides, or it grows, or it stops growing, or it dies. Something like that. There's some sort of effect as a result of it. So let's jump into this conversation. I think you're going to like it. One thing that I want to say about it is it's not so much important to recall, in terms of memorizing the names of hormones. It's great if you can recall them, but it's more of the point is to enjoy them, to understand the bigger picture. And so the word hormone itself comes from a Greek word meaning to excite or to set into motion. So it's kind of like if you think of a big chain of dominoes and you flick it with your finger, that would be the hormone to get going, to set into motion. And so hormones are pretty common in most organisms, and they're produced usually in one part of the plant, and then they target another part, as I was saying before. One of the very first plant hormones that was ever isolated, and it's a pretty cool story, I'm going to actually mention it in this video, has to do with the hormone auxin. And there's actually a couple of different kinds of auxins. It's basically a growth hormone. And when you have auxin, you're capable of normal growth, and when you don't have it, you're sort of stunted, in other words, a dwarf. And so, here you have tall plant, short plant kind of thing. And so the chemical structure for auxin, it's very similar. This is auxin. It's also abbreviated IAA. It's very similar to the amino acid, as you can see here, tryptophan. And so one of the things, I just wanted to show you what we're talking about in terms of chemical structure. Some of the hormones could be steroid-like, and some of them could be peptide-like, for example, auxin. And so just the big picture is that cells can communicate with other cells. It can be locally. Cells can produce chemicals that are secreted from little vesicles, and they target adjacent cells, and this is something known as paracrine signaling. It's sort of local. And then in the nervous system, neurons don't actually come in contact with one another, but they can communicate. How do they do it? Well, in their axon terminal right here, they secrete these chemicals called neurotransmitters, and there's a bunch of neurotransmitters. Acetylcholine happens to be one of them. You should check out neurotransmitters if you're interested in that. It's the way in which neurons communicate. In other words, it's sort of like a post office where you're whispering in one person's ear and then they're whispering the next. So in other words, the brain, for example, can communicate with the foot in order to kick a soccer ball by moving neurotransmitter to cell to cell to cell to cell, and then ultimately the neurotransmitter will connect to the muscle, which through the neuromuscular junction, which is also a synapse, and then it'll cause the muscle cell to contract. So all that is sort of called paracrine. Now, a hormone, for an animal at least, is produced by glands. Now, plants aren't known for having glands, but we have groups of cells that are specialized for secretory purposes, and so these endocrine glands secrete hormones, which are what these blue structures are. And it goes into the bloodstream, and it travels throughout the body, and it goes to a target cell. For example, an endocrine cell could be like, for example, in our brain, in our pituitary, gland, and it produces a hormone that travels in the blood and goes all the way down to the ovary, tells the ovary to start developing a follicle. Or this hormone can come from the adrenal gland, and it goes into the blood, and it causes the lung and heart to increase respiration and heartbeat. And so animals don't have, plants do not have this blood to go into. They have instead vascular tissue, and so hormones can travel through the vascular tissue, in xylem sap and in phloem sap, but it can also be cell to cell as well through simple diffusion. And it's often long distance. We call that endocrine because it's on the inside, as opposed to a gland that's an exocrine gland. Like, for example, your salivary gland produces saliva, which then travels in a duct to the outside, which is your mouth, or the mammary gland to the outside. So hormones, chemical messages. So the research of how plants bend toward the light led to the discovery of plant hormones. It's a really cool story. As you can see, here's the sun, and plants are moving towards the light. And this is an example of what we call phototropism. Tropism means toward something. It's an orientation toward, so it's toward the sun. So in order for that to occur, there has to be some bending, which occurs over on the dark side. So that's kind of interesting. If the dark side cell bends, in other words, the cells elongate, then the whole plant will sort of orientate itself towards the light. And you can actually see the plant moving throughout the day as the sun moves across the sky. It's pretty cool. And so this is what we refer to as phototropism, positive phototropism, moving toward the light. You can even see it in a forest. You know, this is a real powerful event. You know, a forest is very competitive for light, so you got to be able to roll a little bit. And frankly, branching increases the plant's ability to compete. It's going to have more leaves, and it can also move around in order to get sunlight. And so what we know about phototropism and what we know about hormones, the experimental evidence came from this sort of simple corn. When the very first thing that comes out of corn is that radicle, which is the beginning root. But then this very first shoot that comes up, there's a sheath that surrounds the stem called the coleoptile, as you recall that from a previous discussion. It doesn't have to be corn. It's like just grass in general. It's surrounded monocot. It's surrounded by this coleoptile. And if there's, if its light is coming directly above, the shoot will grow straight up. And if the light's off to the side, it'll grow to the side. And so the reason it's bending over here is that there's presumably sun over here shining, which means these cells right here are elongating, which is causing it to bend. Now, what's fascinating, it is a great picture. So here's corn. Look, here's the root system coming off of the corn. This is some sand right here. And you can see it's sort of like stages of development. This first little sheath that surrounds the stem is the coleoptile right there. And then this in red is the very first leaf, true leaf, that's coming off of the corn. But here's the coleoptile. That's what we're talking about. You're like, well, why does the coleoptile, how does that even relate to phototropism? So let's take a look at this. So here's the coleoptile. Again, if light is coming from the left, that means it's going to bend toward it, which means the shaded side cells are going to elongate, which causes it to push over. So it's differential cell growth. That's causing these cells to elongate and to divide and then move in that direction. Something must be weakening that sort of cellulose wall, allowing the cells to get a little bit larger. What's happening? Well, the same thing that's happening in the root. For example, the root is showing positive geotropism, and the shoot is showing negative geotropism. So there's something that's causing, right over here, the shoot to bend, and then ultimately it's going to bend towards the light, and something's causing the root to bend over on this side, which pushes it in the down direction. So in other words, these cells are elongating right here, which is then pushing it down. And these are elongating, which is causing it to move in an upward direction. Well, would you be surprised if I told you that one of the very first scientists to work with phototropism was Charles Darwin himself? And interesting, it's Charles Darwin and Darwin, which is this thing. It's Darwin and his son, Francis. They were both working on this towards the end of Darwin's life. It's pretty cool. He was teaching his son the science. And so what they were doing was growing some seeds, and you can see here's the coleoptile, and they're noticing that this is basically the control. When there's light over here, it's going to bend towards the light. And then they noticed that if they simply severed the tip of the coleoptile, in other words, cut it off with a knife, suddenly the coleoptile, doesn't matter where the candle is placed, it doesn't bend. And so that might lead you to believe that perhaps the tip of the plant is important in some way. I'm not sure if I would know what, but I'm just saying, obviously the tip is very important. Okay. So then they decided to keep the tip in place like the control, and this time cover it up with like a piece of cloth or something opaque. They didn't, I don't think, believe they had aluminum foil. But if you wrap this up in aluminum foil and not allow the light to touch the tip, it still would grow in a straight direction. That's interesting. And then here, it's not so much that you cover the tip. Here's something that's covering the tip, but it's sort of see-through. So clearly you might come to the conclusion that light was important. And then you're like, well, maybe light is important in general. Why just the tip? Well, if you cover the bottom part of the coleoptile lower, again, that doesn't influence it. So it appears to be the tip. So Darwin sort of showed that something about the tip's ability to receive light is very important. But what's curious about that is, I mentioned to you before that you're like, yeah, light, I can see how that's working. But ultimately what's happening is these are the cells that are being affected down below. Do you follow that? If these are the cells that are being affected, the ones that are elongating, which is causing it to grow, what does the tip have to do with it? And how can the tip talk to the side of the coleoptile. So another experiment was conducted in which Boyson and Jensen, kind of a long time ago, 1913, they did what Darwin did. They cut the tip off, and then they put it back on, and they put a little piece of gelatin, so if you can imagine like an agarose gel. Now, agarose gel is permeable. Substances can diffuse through it. So how do you like this? Even though the tip was severed, and you put a gelatin block in between, it still bends. Well, over here, the cut is made, the tip is put back on, and then this time an impenetrable barrier is put there, something called mica. In other words, nothing can penetrate it like for mica, like a countertop. So this is interesting. So this is being exposed to the sun, and nothing's happening. And so this is weird. Both of these were cut off, although this allows things to go through and this doesn't. Any thoughts? And so Charles Darwin, I put this picture here showing phototropism with a dust lamp, and in fact he didn't probably have a dust lamp. He's probably using candle or the real sun. So he was able to determine that the tip is needed for phototropism to take place. And then again, Peter Boyce and Jensen demonstrated that there might be a chemical substance and it might be mobile. And again, you know, where's that coming from? It might be a chemical substance because it needs to travel from the tip to the side for these cells to elongate. And it is capable of penetrating here, but it's not capable of penetrating there. And so this is cool. So then in 1926, Wendt extracted the chemical messenger, so the mysterious chemical message that was coming from the tip, and proposed its name being auxin, meaning to sort of to grow. And so how did this group go about doing that? Well, if you cut off the tip and after it's been exposed to light, and basically you put that onto an agar block, the chemical, now known as auxin, would simply diffuse, like the pink chemical is auxin. Are you grasping that? So this pink is this auxin. So it diffuses into the agar, and then look, here's the agar. So the agar now has the auxin in it. So how do you like that? So over here, when you cut the tip off, nothing. Over here, you put just agar block, nothing. This time, you put the agar block that's been soaking in the auxin, and watch this. Notice how this is dark and this is light. It's diffusing into the coleoptile, and then ultimately it's diffusing down the side here. It's affecting these cells, causing them to elongate and to grow, or over on this side, depending on how you place that. And so if the auxin goes on this side, it's going to bend to the left. If it goes down on this side, it's going to bend to the right. Wow. So it's a chemical, and it was isolated, and it looks like a little tiny tryptophan. It's auxin. That's pretty cool, huh? So that was one of the very first plant hormones that was discovered, auxin. And so it's a classic. And so as you can see, this purple sort of shows that when the auxin travels down on this side, it's going to cause the coleoptile to bend, or phototropism, or geotropism, either one. So plant hormones, that was just the first plant hormone. Plant hormones, what do they do in general? This is the come away message here, this take-home message. They help to coordinate growth. They help to develop plants, in other words, to flower, not to flower, to germinate, for fruits to ripen, to basically do all of these things that a plant needs to do in response to environmental stimuli. Like what we were just talking about, the environmental stimuli was light. So why? Well, again, because plants are planted. They cannot make these adjustments of growth and development by moving. In other words, when winter time is coming and the days are short and it's snowing and there's going to be no more water and you're in a deciduous forest, why don't you just drop your leaves? And so, what's going to cause that? Hormones are going to cause that to occur. Or it's spring, it's time to flower, for example, if you're a flower that blooms in the spring. But hormones are going to coordinate that. And so these are chemical messages to get things done in response to the environment. And so they can control cell growth. They can even control cell death. Sometimes you want cell death to occur. For example, if you're a primary meristem, say you're Procambian, and you're producing xylem cells in the vascular seal of the root, and you want those cells to eventually die, well, a hormone is going to cause those cells to die. And so that's important. Or if you want the plant to grow laterally, it's going to be cell division to sort of push the meristem out, and that's going to be a hormone that causes those cells to divide. And so there's a bunch of plant hormones, let me assure you, but I'm just going to cover a couple of them today that are fairly common in most plants. That's why I'm choosing these. So auxin, some cytokinins, you might even be able to predict what that does, some gibberellins, and some abscisic acid and ethylene gas. And so one thing about plant hormones is that you really just need a small amount. All you need is a little like knock on the door and the cell knows what to do. So they're needed in relatively small amounts. And so plants are needed in small amounts of hormone, and it's not even so much, like a lot of cool experiments were done in terms of like too little hormone, too much hormone, and basically one of the major factors is the ratio of the hormones to each other that really cause an effect. And so this is the IUPAC name for auxin. Auxin is sort of the common name. So this is IAA, which is the hormone that is involved in phototropism and geotropism. And it's, as what we know, it's produced in the top of the plant, in the coleoptile tip. And so there it is. That's the chemical structure of it. And so auxin affects many different aspects of the plant development, but chiefly it stimulates elongation and the stem and growth, as well as phototropism and geotropism. So it's a growth kind of hormone. So what's interesting is developing seeds can produce auxin as well. So that's the source of it. And it promotes the growth of the fruit around it. And since we discovered that, we're able to induce tomato lines, for example, to develop seedless tomatoes because the synthesized auxin that we're spraying on it substitutes for the normal synthesized auxin by the developing seeds. I find that's pretty interesting. And then these cytokinins, they, as the name implies, they stimulate cytokinesis or cell division. So cells are growing. So if you want a cell to grow, it's going to take a cytokinin to do it. And so this is a bit of a detail, but I find this kind of interesting. And so despite a lot of effort, the enzyme that produces cytokinins has not been purified. And the gene for that hormone has not been identified as of yet. And so it might even be that plants themselves do not produce the hormone, but rather bacteria that live symbiotically with them produce it. Isn't that crazy? And so along those lines, many years ago, when individuals in Asia were growing rice, they noticed that there was something weird was happening to certain crops of rice. Some of the rice were growing so quickly and so long, the stems were growing so long that they actually topple over because of the weight, even before they were able to mature and flower. And so it was discovered that the reason they were growing so fast, sort of like foolish seedlings, is because there was a fungus that was infecting them. And as it turns out, that this hyper stem elongation was caused by the hormone gibberellin, which causes shoot elongation. So if you're familiar with like lettuce, which tends to be like a low growing, meaning like close to the soil kind of thing, like a head of lettuce, I did an experiment once back in college where I was adding different concentrations of durabellic acid, which is another way of saying durabellic acid, and varying the concentration. I was able to get the lettuce seedlings to really elongate. That's pretty cool. So it affects stem elongation, so it's a growth kind of hormone like auxin. So that's kind of neat. We can also spray gibberellins on grapes, which cause them to grow a little bit larger and be all puffy like this. And it's like not only are they desirable to be purchased this way, but it also enhances air circulation between them, and so it makes it, when it rains, or if you're just simply watering your vine, water tends to, when the grapes are small, get inside, and then it causes mold to grow. And so it helps when the grapes are a little bit larger. And then an embryo of the seed is rich in gibberellins as well. And so after water comes into a seed, it imbibes seed after hydration. There's a big release of that hormone from the embryo. And what that does is it causes the seed to sort of break its dormancy and germinate. And so that hormone tells the seed it's time to break. And so this, as you can see here, the root is coming. Here's the root hairs. So it's kind of neat. And then citric acid. I always think of, when I say that, probably pronouncing it as citric acid. It reminds me of a pair of scissors, which is kind of a cutting thing. And cutting means like for cells to sort of stop growing. So this is like, there's a hormone like auxin and gibberellic acid, and cytokinins are all kind of growth. This is like not growth. Like when you're cutting something, stop growth. And so it's kind of antagonistic to a growth hormone. So it stops growth. And so what's the effect of it? Why would you want to stop growth? Well, seed dormancy. I mean, like, remember we said that when the seed is, the zygote is fertilized and it forms the embryo, and then suddenly the seed becomes dormant? Well, you want seed dormancy. And it's like, well, how do you know that there's this hormone inside of a seed? Well, the concentration of it is very high inside seeds. And so what's interesting is being dormant or not growing is a survival value, because you do not want the seed to grow unless the environmental conditions are right. And so when the light is better, or if it's warmer, or if it's more moist, then you would want germination to take place. And so it's kind of a trade-off between, gibberellin and citric acid. And then here's a really cool hormone. I like this one a lot. It might be my favorite. You've heard of the expression, it only takes one rotten apple to spoil the bunch. And the reason that is, it's kind of like, that must mean that one ripening fruit affects others. And how does it do that? Well, it produces a gas. This particular hormone is gaseous, which means that it's volatile. It can actually diffuse in air. And so fruit ripening, for example, if there's a rotten apple in the bunch, it really will cause the other fruits to ripen at the same time. It triggers it. And so even on a tree, once one apple starts to ripen, all of them start to ripen. And again, that's really important. Ripening fruit is critical because it's very attractive to animals to come along and help for seed dispersal. And so it's kind of a cool example of a positive feedback in hormones. Usually when a hormone is produced and the concentration gets too high, the organism stops producing the hormone. Does that make sense? It sort of regulates it. It's called negative feedback. This is positive feedback, which is kind of rare. In other words, the more ethylene, the more ethylene. And then it ripens, and then even more ethylene, and then even more ethylene. And so how does it ever stop when all the fruits are ripe? And then they're either picked or they fall off. Another example of positive feedback is during labor. There's a hormone called oxytocin. Now, plants don't have labor. We're talking about female mammals now. There's a hormone called oxytocin that increases labor contractions in the uterus, and then that sends a signal to the brain, to the posterior pituitary gland that makes even more, and then even more and even more, and it'll never stop. Well, when the baby comes out, then it stops. And so fruit can be ripens very quickly. Like, for example, if you buy some bananas and you want them to ripen quickly, you can put it in a bag. Or if you can put your bananas in a bag, or apples in a bag, or fruit in a bag in general. Now, this may seem trivial, but boy, sometimes, if you're in agriculture and you're shipping or flying in an airplane, like several tons of bananas from Central America or South America to the United States, it is no game. You don't want those bananas, you don't want a ripe banana inside there, inside the cargo hold of the plane. It could cause all of the bananas to become ripen too quickly. You lose a lot of money. And so we've learned that, like, flushing with carbon dioxide prevents the ethylene from accumulating. And so there's certain chambers on ships, on trains, in trucks, and in airplanes that circulate to prevent ethylene gas from accumulating. How about that? And then coming to an end here, I want to talk a little bit about programmed cell death, something called apoptosis. And so cells, sometimes you want plant cells to just simply die. And so they're not programmed to die on a particular schedule. They don't shut down. So if you want a plant cell to die, what we think is happening is that there's an active expression of a gene which produces an enzyme in the cell that basically it's sort of suicidal. The enzyme comes along and breaks down crucial things like chlorophyll, DNA, RNA, proteins, the membrane, and then the cell dies. So it takes an active role in death. And it's like, well, why would you want that? Well, some examples of this is that you want your leaves to fall in autumn. So in other words, the petiole, the cells that are holding the petiole to the stem, those cells have to die. And then the petiole becomes weak, and then the wind kind of blows it off. Or it's a cue to the plant to die, an annual plant, after it's flowered in the first season. Or if you're xylem, and you want your xylem cells right over here to die, then this is what happens. And so a great example of programmed cell death, or apoptosis, is the leaves falling in autumn. And so it's a great adaptation to have your leaves come off like this, because basically if there's not a lot of sun, all you're doing is losing water, and it's a lot of work for no good. And so this is cool. This is showing that right at the petiole, this is where the petiole is connected to the stem, right down the line, it's almost like perforated paper. All of these cells are programmed to die right there. And so it becomes very, 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 very thin. And it weakens, these enzymes that I'm talking about weaken the cell walls, and therefore just simply wind will come and knock it over. And so I hope you enjoyed that. I know plant hormones, sometimes people love them, some people don't like them, but I think it's just a matter of getting to know them and appreciating some of the cool things that they do. So I hope you enjoyed it, and I hope it was helpful. Thanks for watching. Everyone, it's Mr. Sinti, and today I want to talk to you about primary production. It's basically a conversation about photosynthesis, and the truth is energy transfer pretty much underlines all biological interactions, and so it's rather important, and I might add somewhat interesting. And so primary production is basically, we're talking about the conversion of the energy of the sun, especially if we're going to consider photosynthesis. And let me jump back to that. And so let me throw in a little sun right here. And so what we're talking about is the conversion of the energy of the sun into carbohydrate. And so carbohydrate, plants are able to produce a carbohydrate, CH2O. And how are they able to do that? They're able to take in carbon dioxide from the air and water from the soil. And then as a consequence, they also produce oxygen as well. And so this is called primary productivity, because what we're talking about is these sugars, these stored organic energy sinks, are going to be what is powering the trophic level. In other words, these sheep gently grazing right here in this grass are benefiting from primary productivity. So the plants, the autotrophs, the photosynthesizers are producing that sugar, and that's what's powering all the trophic levels up. And so this is happening both on the land and in the water. And so in this aquatic environment, let me go back here. In this aquatic environment, it's interesting because here you can see algae, and the light is traveling down into the water, especially in the photic zone, and the algae is picking it up and it's converting it into carbohydrates and in oxygen. And so let me actually take a side look here and sort of diagram what's happening. I think it's important to consider this. And I'm going to, so picture that creek that we were just looking at, okay? And so I'm going to attempt to draw that river. So here we are underwater, this is up above, and I'll throw in the sun up here. It's shining into the water. Okay. And so this is, again, not very deep. This is the photic zone. And then in the water, here are the algae or the aquatic plants. I'll just make a big leaf like that. And so what's happening is this plant, or algae, what have you, photosynthetic organism, is producing carbohydrates and oxygen. And how they're able to do that is that there's dissolved carbon dioxide in the water, and the carbon dioxide is coming into the leaf. And in the leaf, there's little tiny organelles called chloroplasts. And this isn't a discussion per se of this, but it's basically where photosynthesis is taking place. Chlorophyll is the pigment. And so it's taken in water, carbon dioxide, right there. These are the inputs to photosynthesis. And through the chemical reaction of photosynthesis, the plant is making a carbohydrate. And there it is, carbohydrate is also producing oxygen. And so there you have it, photosynthesis, primary production. And so the interesting thing is, let me pose this question, like, why are we even interested in it? Well, we're interested, again, as I said, because it's very important. For example, if you're a consumer in the water, you need to make sure that there's enough plants to eat, and there's enough oxygen in the water in order to survive. And likewise, this is important on the land as well. And so one of the obstacles in studying primary productivity is that it's very difficult to measure just how much carbohydrate and oxygen plants are making. Because, as it turns out, they're actually consuming some of the sugars and oxygens themselves. Now, this may be an obvious statement, but I'm going to point it out. Because inside of plants are capable of respiring. In other words, cell respiration. And they do this in another organelle called the mitochondria. So let me write that out. So cell respiration. So the reason that they're actually producing carbohydrate and oxygen is so that they can burn it. And so the sugar and the oxygen is burned in cell respiration, and the outputs of cell respiration is carbon dioxide and water. Fancy that. It's basically the cycle, the energy cycle. Okay, so what we're talking about, the total production, the green right here, sugar and oxygen, what we call that is the gross primary production, or GPP, gross primary production. But it's very difficult to measure that because, as you can see, they're consuming that. And so there's still some sugar left over, and there's still some oxygen left over. So let me go with that. So check this out. The leftover oxygen diffuses out and is in the water. And so this is leftover minus respiration. And so if you were to calculate this, let's just go let's go white up here. So we could say that plants' gross primary productivity, in other words, the total amount of carbohydrate and oxygen that's produced, is really the result of, if you were to look at that gross primary productivity, you're like, well, what is this? Well, in fact, respiration is using some of it. Do you follow that? It's using some of it. And so what is available is something called net primary production. In other words, this is what you could directly measure. If you're standing over here and you're sampling the water, you can measure net primary productivity. To know how much respiration is going to take place. And so we can solve this problem, actually, but I just wanted to point out that it's a little bit of a dilemma, that we're only able to directly measure net primary productivity. But if we can determine how much they're consuming during cell respiration, then we can determine gross primary productivity as well. So I just wanted to point that out. So let's go back over here, if we can, to the slideshow. So all that's happening in the river, there's obviously the fish can eat the algae and getting some sugar, and also the dissolved oxygen, they're breathing it. And so we have these two ideas, gross and net primary production. If I can use an analogy, it's sort of like if you've ever had a job, let's say, for example, in the summer, and your salary is, let's say, $1,500 a week. And so you're like, well, that's your gross salary. But in fact, taxes are going to be taking some money out of that before you can actually get the $1,500. Say that taxes are $500. So what you're actually taking home, the take-home salary, is $1,000. So this is what I'm talking about. The net primary production is the $1,000. And the only thing you can measure is the $1,000. You're like, hey, look, I have a check for $1,000. And you're like, well, interesting. Since taxes took out $500, I guess I originally made $1,500. So that would be the gross. And so this is our sort of our logic here. So the primary production is the total amount of organic energy that is produced in photosynthesis and oxygen. It's all the total amount of light energy that's converted to chemical energy. But the net is basically equal to the gross minus the respiration. So in other words, if this is your $1,500 minus your $500 for taxes, then you're actually taking home $1,000. So we're only able to measure net primary productivity. But we could determine what the respiration rate is rather cleverly. So let's take a look at this. And so check this out. So say that this is a river in question, and we're kind of curious about primary productivity in the river. Okay, so this happens to be the Merced River in Yosemite Valley. And so since oxygen is the more easier thing to measure, we have the ability to use sort of some chemical means. Like we're able to actually stick these little ampules into the water and sample it, and then do some interesting chemical reactions that will actually turn what is invisible, dissolved oxygen, into a color. And for example, in this picture here, you can see the darker the blue, the more oxygen is in the water. And there's different chemical tests that you can employ into this river. You could use chemicals, you can even use probes. You can stick a probe, electronic probe in there, and actually read it off of a sensor. It's pretty cool, but let's safely say that you can measure dissolved oxygen in the water. And so when you're doing that, it's a little bit easy to be able to measure it, but you can't measure gross primary productivity because, again, they're consuming some of that oxygen themselves during respiration. So what you're actually needing to do, so let's take a look at this. So net primary productivity is actually what's available in the river. We can measure that. So the idea is like, well, what's the gross? So in order to know what the gross is, you have to know how much they're respiring, how much of it they're actually using up. And the only way that you can determine that is, check this out, it's rather clever. You could take some of the plant material, in other words, sample the water, and let me try to draw this. So you could scoop up the water and put it in you can put it in a container like this. and put it in a water bottle like that. Okay, so here it is, and inside of it, there's obviously some plant material like this, and you can measure the amount of oxygen in the water. But check this out: if you took the same bottle and you put some aluminum foil around it, or put it in some sort of opaque material around it, in other words, you're blocking off the sunlight. So if you block the sun, so there's no light penetrating into the bottle, what you can do over a set period of time, let's just say it's 1 day, what you could do is you could initially determine the amount of dissolved oxygen in the water. Okay? So let's just make up some numbers. Let's say that initially the amount of oxygen in the water was 10 parts per million. There's the units, parts per million initially. And then after 1 day, since they're not producing any more oxygen because you've darkened the bottle, they're only respiring. So in 1 day, you sample the water again, and guess what? There's less. Predictably, there's 8 parts per million. And so, interesting. So the plants are respiring. So how much did they respire in 1 day? That would be 2 parts per million. So if the respiration is 2 parts per million, and you originally measured 10, that probably means that, the gross primary productivity is in fact 12. So it was making 12, using 2, therefore 10 is available, and that's the net. 10 is available. Available for what? Available for fish. Well, I say fish, but really any consumer. And so photosynthesis and respiration play an important role in primary productivity. Okay, but there's some other, those are biotic factors. There's some other factors as well that contribute to the amount of oxygen in the water, and this has to be considered. There's some abiotic factors in addition to photosynthesis and respiration. So this may be just totally obvious. Again, a person on the street, if you ask them, hey, what's influencing oxygen in the water? And they're like, the first thing they might say is, well, look at this. The water is turbulent. It's rushing over these rocks in this sort of ripple zone right here of the river. And so waterfalls and wave action introduce oxygen into the water. And so that's a physical phenomenon that it's windy and the rapids introduce high levels of oxygen. They may not mention photosynthesis and respiration, but this is a physical factor. So in other words, turbulence and winds. And let me throw in a couple other ones. You may not be aware of the fact that temperature, and this could be experimentally observed in the lab, and so you can literally look at the effect of temperature on how well the water holds on to dissolved oxygen. When I say dissolved oxygen, I don't mean H2O. I mean molecular oxygen. I'm talking about like this. I'm talking about O2 in the water. Think of it as like dissolving sugar into the water. You're dissolving oxygen in the water. How about this? Colder water holds more oxygen than warmer water. So cold water, you can look at this. So if it's colder in the water, there's going to be more oxygen. And so obviously I mentioned this in detail. Photosynthesis, photosynthetic activity, if there's lots of light shining into an area, you can expect more oxygen to be produced, because that's what's producing it, is the energy from the sun. Now, decomposition, this is a curious one. So as organic material like sugars decay, if there's too much decomposition, in other words, there's a lot of bacteria in the water, decomposing, in other words, breaking down the algae. As it turns out, the bacteria, let me see if I can draw that in there. The bacteria, if there's a lot of bacteria in the water, and this could be a problem, they're not only breaking down the sugar, but they're respiring. Bacteria are not photosynthesizing, so they're actually consuming the oxygen and the sugar in the plants, and so they're reducing the oxygen. So if there's a lot of algae decomposition in the creek or in the lake, the dissolved oxygen should be going down, because they consume it. So that's a concern. If there's a lot of algae bloom, then ultimately the dissolved oxygen will go down. And you're like, again, you need to come in with the attitude that this is important to care about, meaning that, okay, dissolved oxygen in the river, whatever. But no, the fish are going to die, and so that's fairly critical. And so salinity, how about this? This is, again, just a physical factor, the fact that salinity affects the amount of dissolved oxygen that the water can hold. So fresh water holds more oxygen than does salt water. So as the water becomes more and more salty, more and more warm, more and more bacteria, the amount of dissolved oxygen will go down proportionally. And so these are abiotic factors. And so, again, you can go out here to the river, and you can say, whoa, look at this. It's kind of windy, and the water's rushing over these rocks. It looks like it's pretty oxygenated. And so, how do you know this? Well, again, you can stand over here along the rock, and you can sample the water and try to determine the net primary productivity and see how much oxygen is available. And so that's of consideration. And so when you look at, beyond just a river, and you're looking at the Earth in terms of primary productivity, where's the most primary productivity? In other words, think about it right now. Where is the most oxygen being produced on the Earth? And if you answered the ocean, you would be correct. Where's the most sugar that's being produced on the Earth? Where is it? It's in the ocean. And so ecologists are, it's difficult to grasp this number, but ecologists believe that something like 150 billion metric tons of organic material are stored every year on the Earth. And most of that photosynthetic primary productivity occurs, as you can see here, percent of the Earth's net primary production is occurring in the open ocean because it's so vast. Look at this, the percentage of the Earth's surface. Look at this, 65%. And so far it would shoot off, and that's why there's a little break right there. So the ocean is where the most primary productivity is taking place. But the truth is, if you look at just a small area of it, it doesn't really produce so much. But because it's such a huge area, it produces quite a lot. And then of second importance is the tropical rainforest. There's a lot of primary productivity in the rainforest as well. And so that's a real productive area. And so that's predictably, if there's a lot of carbohydrate and oxygen coming out of the tropical rainforest, that must be able to sustain a lot of higher trophic levels, and so you get a lot of species diversity. That's a real important area. And so what's fascinating about that is not only species diversity, is that it doesn't occupy so much of a large area of the Earth, so which makes it very valuable since it's rare. And so just to sort of point out, go back to the water for a second, just wanted to point out that all this primary productivity is happening up here in the photic zone. So because it's being powered by light. So oxygen is being produced up here, and sugar. So most of the energy transfer is up here in the photic zone. And so one of the things that can limit primary productivity is nutrient, because let me go back to this picture. Plants, this is kind of a weird thing to say, but plants for the most part prefer being on the land because the soil has a lot more nitrogen, a lot more phosphorus, there's a lot more carbon dioxide, you get a lot more sunlight. Although it's dangerous because you can dry out on the land. But if you're in the water, sure, you get lots of life in the sun, and there's a lot of carbon dioxide dissolved in the water. So some of the things that are limiting primary productivity are other nutrients, like, for example, nitrogen and phosphorus are elements that can limit primary productivity in the ocean. And even something that you may not think about is iron. I think you know that iron, we need iron. You might know that you need to be able to eat iron. You're like, why do I need to eat iron? One of the reasons is that it's important for our red blood cells to have that in order to carry oxygen in the body, and we also need iron for more subtle things like electron transport system and cell respiration, but we need iron. So plants and algae and phytoplankton, they also need iron. And so how about this? So if nitrogen is limited in the water, there's not going to be a lot of algae. And if phosphorus is limited, and iron is limited. And so you can conduct experiments and actually add these elements into the water to see if there's an increase in primary productivity and net productivity. And so sometimes you don't want these limiting reagents or limiting elements in the water. For example, as I was talking to you about it a second ago, decomposition of algae can be detrimental, because if there's a lot of algae that's booming, then when they all die, it's going to actually decrease the amount of oxygen. And so some of our favorite lakes, like, for example, Lake Tahoe in California, you might have seen bumper stickers around saying, keep Lake Tahoe blue. In order to keep it blue, you have to limit, you can't have sewage getting into the lake, you can't have fertilizer from outlining farms getting, and you can't have nitrogen and phosphorus and all of this, like soaps, running into the water. Because what that's going to do is increase the algae, and then it's going to die, and then it's going to, decomposers are going to consume the oxygen in the water, and ultimately dissolved oxygen is going to be less, and therefore it could hurt the whole lake in general. It'll be dominated. Now, has this occurred? Have we ever seen evidence? Yes, it has. It's called eutrophication. Again, it's when high concentrations of things like nitrogen, phosphorus. How do they get into the water? Well, we like to farm next to lakes because that's a great source of water. And so fertilizers can leach, in other words, run off excess into the water source, and it can cause an excessive algae bloom. And again, I pointed this out in another video. Maybe you saw it, maybe you didn't. But this process called eutrophication is happening in one of California's most prestigious lakes. And it's rather ironic, and it's rather brutally sad, if I might add that, is that the lake is called Clear Lake. And it's not clear. I mean, you got to think back in the day, it was probably pretty clear, but now it's not. You can see it right here. It's green in coloration. There's a tremendous amount of algae. And it's clearly the effect of human interaction. It's like the farming that is along the lake has allowed these limiting reagents to come in, and then the algae blooms, and then it's an unnatural increase in phosphorus in the lake, which then causes the dissolved oxygen ultimately to be reduced. So you can see the algae growing right here. And so after a period, the algae dies, falls to the bottom, it's decomposed, and ultimately it lowers the oxygen in the water. So oxygen in the water is rather critical, because then the fish start to die, and then it sort of goes down this spiraling vortex of trouble. And so ultimately, we can study this. We can hope to control the amount of pollution, which then ultimately controls these things called algae blooms and eutrophication. And studies have been conducted, even on a large scale. It's hard to study this on a large scale, but you can sort of block off two big bodies of water, these big lakes, and you can see what the effects are of adding nitrogen and phosphorus. And over here, nitrogen and phosphorus were not added. And so it's a fascinating process. But primary productivity, again, underlies most of the important biological interactions in ecosystems. So I hope you enjoyed that brief look at primary productivity. Thanks for watching. Hi, everyone. It's Mr. Sinti, and thanks for tuning in. Today I'd like to talk to you about how plants can respond to the environment. And in particular, they respond to the environment in this discussion, to the light, in something called photoperiodism. And just the bigger idea before we go into detail, the bigger idea is that plants, in order to survive, have to be sort of cognizant, if you will, sort of metaphorically cognizant of time passages. So in other words, like seasons passing, temperature, these sorts of things are really crucial, because it'll affect when's the best time for certain plants to flower. So as you may be familiar, plants can flower. Some plants flower in some seasons, some do in other seasons. And this is a discussion today about how plants can determine when to flower. And in a previous video, we talked about when seeds will germinate, but it's basically this. Well, let's get into it. Let me just click over here. So there's an appropriate appearance of seasons. Seasonal events are critical in the importance of a plant. And so plants respond. And this is what it's all about. This is they adjust their physiological responses. In other words, their flowering or their seed germination based upon environmental stimulus. And what's the most important environmental stimulus for seasons changing? It's going to be the light. And so this is called a photoperiod. It's the relative length of day and night. And you're like, well, wait a minute. Is that really the case? I thought the length of the day and the length of the night was the same. Now, in fact, it does change. And so the midway point of a 24-hour day is 12.